Hi everyone, I'm Austin Montgomery. I'm an MD PhD student in Amanda Nelson's lab at Penn State College of Medicine. And today I'm going to discuss how we use spatial transcriptomics to discover how different histological features of lesions in hidradenitis suppurativa compare to one another. So hidradenitis suppurativa, or HS, is a chronic inflammatory skin condition of follicular occlusion which can lead to abscesses, draining skin tunnels, and fibrotic scars. HS is not rare, with approximately 1% of European and US population suffering from the disease. Many patients are unsuccessful in treating their HS. Approved therapies work in less than half of patients. So there's a need to characterize HS pathogenesis so we can better develop treatments for HS patients. So unbiased approaches like RNA-seq can identify novel pathways, targets, and cell types in HS. Well, spatial transcriptomics specifically permits comparison of HS histological features that may be difficult to isolate, such as tunnels. And with this in mind, we use spatial transcriptomics to understand how HS lesions develop and progress. Now, to perform our spatial transcriptomics, we sectioned HS samples from 4 different patients, and each of the 4 samples were Hurley stage 3 from the axilla and showed typical features of HS, such as epidermal hyperplasia, an inflammatory infiltrate, and fibrosis. Now, 1 sample contained a tunnel, and 2 of our samples contained inflamed follicular structures. With the help of a dermatopathologist at Penn State, Dr. Matt Helm, we manually annotated each of the spots and each of the features in our HS samples, so we could do further analysis with the associated RNA-seq data. So spatial transcriptomics is a special RNA-seq method. And RNA-seq traditionally measures what genes are most actively transcribed. So traditional bulk RNA sequencing, if we compare it to a fruit tart, it would be like if you took all the fruits from the fruit tart, mixed them up in a blender, and made a smoothie. Now you could know what fruits were from the fruit tart, maybe you can even taste them, but you wouldn't know where they were on the fruit tart originally and how they were related to the other fruits. So that's kind of like bulk RNA sequencing. You can figure out what genes are there, but you don't know where they were and how they connected with one another. Now, single-cell RNA sequencing, that retains the single-cell information. So if you took that fruit tart, pulled the fruits off of it, and then organized them into a fruit tray, that's kind of single-cell, where you can know each of the individual fruits that were there, but you've lost the spatial information, and so you don't know how those fruits connect with one another. And then spatial transcriptomics is just like a fruit tart. You get to know where each of the fruit was or where each gene is in relation to one another and how that correlates with the different structures on histology. Now, in accordance with our fruit tart analogy, spatial transcriptomics can map active genes and regions in histology. And as proof, we would expect increased keratin expression in keratinocytes in this tissue section. And as evidenced by these bright spots here in the epidermis, we do see increased keratin 10 expression in the epidermis. And there's little expression in the stroma. So if we zoom into the cellular level, we see that there's increased keratin 10 in the suprabasal keratinocytes and not in the stroma. And the gene expression distribution gets across the HS features, kind of like this, gave us confidence in the biological validity of our spatial transcriptomics. So next we analyze how the HS histological features compare to each other. Specifically, we wanted to know how an HS tunnel compares to the overlying epidermis. And so we performed differential expression with DE-seq2 of all of the brown spots here in the tunnel and compared that to the aggregate expression of all of the orange spots in the overlying epidermis. And differential expression showed increased expression of genes in the tunnel that have previously been associated with wounding, like lipocalin-2 and the kallikrines, and those have also been previously shown to be increased in HS. Now, gene set enrichment analysis of the increased genes in the tunnel showed enrichment in epidermal cell differentiation and development, as well as a response to bacterium. And that makes sense because there's this idea of the tunnel as an inflamed proliferative epithelial structure. So we next wanted to know if these tunnel upregulator genes were specific to a skin signature or if they were enriched in other types of tissue. So we did an enrichment analysis against all of the mRNA expression data of the protein atlas, and we showed that there was much more of an enrichment of internal epithelial structures compared to the epidermis, such as the GI tract and the female reproductive tract. Now specifically, the tunnel upregulated genes were more than 20 times enriched in the esophagus compared to the epidermis. Now, keratin 13 was the gene that was our first tip-off here. Keratin 13 is a differentiation suprabasal keratinocyte marker, typical in non-cornifying epithelia, so kind of like the oral mucosa and the rest of the GI tract. And so we looked at keratin 13 across our 4 HS samples, and we showed, of course, that it's in the tunnel since we identified it with our differential expression. We also showed it in another inflamed follicular structure, but it was absent from our 2 other samples, and it wasn't in any of the healthy samples that I looked at from a different data set. Now this is interesting because keratin 13 has actually been shown to be dysregulated in some other previous HS studies. So this is very preliminary data, and I did this last week, so don't ask me too many questions about it. But we did immunohistochemistry of keratin 13 on some healthy samples and on some HS samples, and we showed that keratin 13 was negative in our healthy samples in the epidermis, but it did show up in the HS tunnel as well as the invaginating epidermis. And we saw that across a couple different samples. And so this is preliminary. I still need to optimize the staining protocol, and eventually we want to do a bigger cohort of these HS samples so that we can know if this keratin 13 is in all tunnels or if it's in a specific subset of them. So the key takeaways is that these HS tunnels have a mucosal signature, and it seems to be more similar to a digestive or the female reproductive tract than maybe to the epidermis. Additionally, keratin 13 is in the HS tunnels, but it's not in the healthy epidermis. So this is a unique, maybe biomarker of the tunnels of HS. And our biggest question is, can we find therapies based on the drivers of this mucosal signature? Is this a response to inflammation? Is this driving HS, or is it trying to downregulate HS? We're not sure. But hopefully we can find something in the near future, one that is driving the mucosal signature, and also then can we see if that will help develop treatments for patients.